we're going to talk about microscopy and staining. So this is tools of the trade, tools that we use in studying microbiology. Hopefully, most of this material will be replicating and perhaps going a little bit more in depth some of the stuff that you've talked about in lab. Is anyone here not in lab? A bunch of you aren't in lab. Has anyone here never taken a microbiology lab? Same people. Okay, because I know sometimes some people like pass the lab, but they want to retake the course over, and I just figured I would check. All right, you guys are going to want to take especially good notes for this because I'm going a little bit quick over some of this material with the assumption that people are sort of familiar with microscopes. Microscopy. Microscopes are the first, discovery of really good microscopes was what kicked off the science of microbiology, and microscopes are still to this day the most important tools that we use in microbiology. There are 3 properties of microscopes that define sort of how good a microscope is. The first one is magnification. Magnification is very intuitive. We all have a good guess as to what magnification means. It means what you think it means. It's how big something looks compared to how big it actually is. So if it looks 10 times bigger than it really is, that's a 10x or 10 times magnification. Magnification by itself is not always very helpful. As a lot of you will be familiar with from, snapping pictures with your camera or your phone or whatnot, and you can take a picture of something that's far away, and then you can go in and zoom right in on that area and blow it up so that it's huge, and it will be greatly magnified. It will also be crap. It will be totally pixelated, blurry, big, but not very useful. So magnification in and of itself is not always a super useful thing if it's not accompanied in an increase in the other attributes that we talk about as well. Magnification is usually controlled by the type of microscope that you're using and the type of lens that you're using. The second big point for microscopes is resolution. Resolution is basically the ability to tell that 2 things are 2 things and not 1 thing, which sounds a little bit confusing. But let's take a look at it here. So if I put a dot here and a dot there, y'all can tell that that's 2 dots, right? No problems. If I put a dot here and a dot there, you can all tell that it's 2 dots, right? Okay, what if I put a dot here and a dot there? Who can tell me with surety whether that's 1 dot or 2 dots? Raise your hand. You got a couple of people with really good eyes. So I get about here, and I can no longer tell if it's 1 sort of funky shaped dot or if it's 2 dots side by side. I can't tell whether or not, I see that it's a weird shaped dot, but I can't tell if it's 1 object or 2 objects from this distance. You get closer, and you go, oh yeah, that's definitely 2 dots. I can see that. So that's resolution. The ability to distinguish that 2 separate things are separate things and not the same thing. This is controlled by a bunch of different factors. Some of them, like the lens quality, better lenses are going to have better resolution. Better light sources are going to have better resolution. Better whatevers are going to have, better microscopes, better put together microscopes that are more stable. Lower temperatures can often affect resolution, but ultimately, even if you have the best, if you assume that all of the quality is good, that you've built the best possible this type of microscope that you can build, resolution is still limited, and it's limited by the type of microscope you have and what you're really looking at, light or electrons or atomic force or whatever. So there's an upper limit on resolution. The third principle, the third property is contrast. Contrast is the ability to distinguish an object from the background, or the ability to distinguish two objects from each other. There are some microscopes that can increase contrast, but the primary way that contrast is achieved is through staining. And we'll talk about staining in the second half. So who here has seen like one of your teachers give a PowerPoint presentation, someone give a PowerPoint presentation, and it's like, it's got a black background and dark blue text? Or it's got a white background and like yellow text, and you look at it and you go, the heck? Does that say something? What made that person think that was a good idea? That's contrast. That's poor contrast. Here is an example of good contrast. You got black on white or white on black. It's very easy to tell the difference there. Here's the problem. Most cells, and what color are most cells? Anyone have a notion? Clear, right. So, yeah, they're actually, there's a little bit of stuff in there. So they're going to be a very light gray. So often you're looking at a cell that's very light gray on a white background. How much contrast is there? Not very much. So you're going to want to do something to increase the contrast. So this is sort of, this picture's from your book. This is a representation of different things that you might want to look at with a microscope that are most of them, relevant to microbiology and what you would use to visualize them. So the human eye at its best, like really close up, staring right at it, and if you have perfect vision, can go down to seeing something that's about 1/10 of a millimeter at best. So you can actually see some of the biggest microbes with your naked eye if you're really close and if you have really good eyesight. And you can't see much detail, but you'll see like little flecks of something floating around. So things like eggs, bugs, and really, really large cells are all visible to the human eye. Compound light microscopes, or light microscopes, like really any type of light microscope, is going to be useful on, I actually see people look at things that are about 2 or 3 inches long with certain types of dissecting scopes, all the way down to about half a micron. Your average bacterial cell is about 1 micron. So this is a bacterial cell. Half a micron is about the best that you can look at with a light microscope under any circumstances. And there's a reason for this, and that reason is light microscopes, you're looking at light. And a lot of you might not have, thought about this in the past, but light has a size. Visible light runs from like 400 nanometers up to like 600 or 700 nanometers. I don't remember off the top of my head, but it's in that range. It's in that range of about half a micron. Like the smallest light that you can see is about half a micron. And you can't use light to see anything that's smaller than light. Or at least not to resolve something that's smaller than light. You can see single molecules if they're fluorescent, but you can't tell where in that cell, where in that half micron area they originate from, because the light itself is that big. So compound light microscopes can go all the way from like fleas and insects, through protozoa, human cells, organelles, down to bacteria. The smallest bacteria are right at the edge of what you can see with the compound light microscope. Scanning electron microscopes, I've seen like pictures of bugs, insects. You get really nice detailed facial maps of creepy little crawly things. At the top end, all the way down at the bottom end to things that are, you know, amino acids, proteins, DNA, certainly viruses.
I've used a dissection microscope before. Which one's that?
Dissection microscope is actually not a compound light microscope. It's a single lens light microscope. And it's going to go from about
Look at bugs
here to about there. It can't go quite as far as a compound light, but that's what you would use to look at things that are bigger than a centimeter. Like I used to look at flowers, basically, plant reproductive organs, when I was doing plant breeding through dissection scope. TEM, transmission electron microscopy, has a little bit more resolving power than SEM, and tops out a little bit short as well, but covers basically the same range because you're using the same thing, electrons, to look at stuff. Scanning tunneling microscopy, this is actually measuring quantum effects. Quantum effects which happen over very, very small distances, like 10 nanometers, which is the size of like a ribosome or a protein, much, much smaller than a cell, or even the smallest organelles. That's big. That's so big a scanning tunneling microscope can barely see it. It's huge field of view. Scanning tunneling microscopes can go down to visualizing single atoms and below. The guys at IBM and you know, the Lawrence Berkeley National Labs and a bunch of these high-power places that can use these things to like actually pick up single atoms and move them around. So like I saw a paper where they picked up single fluorine atoms and they would move them so that when they looked at these single atoms, it spelled out IBM in like 14 atoms, something like that. It was kind of really cool. So light microscopy breaks down into a few different types of light microscopy. There's bright field. This is what most of you are familiar with. You're looking at a bright field with a dark sample. You're not actually looking at the sample exactly. What you're looking at is the sample's shadow. You're shining light up at your eyes, and when it passes through the sample, it casts a shadow. And what you see is that shadow. So it depends upon the ability of your sample to be able to block out some of the light. So it can contain a single magnifying lens, if it's a simple microscope as opposed to a compound one. It's basically a glorified magnifying glass. These were the first type of microscopes that were invented. Compound microscopes are what you guys are more familiar with, what almost everyone uses nowadays. They have two lenses, an objective lens which is near your eyes, an objective lens which is near the sample, and an ocular lens which is near your eyes. And so the light gets magnified twice. And you multiply those two numbers together to get the total magnification. So if you're looking at 20x magnification on the objective and 10x magnification on the ocular, what's the total? 200 total, right. Many of these are going to use oil immersion to increase the resolution. Just to show you how oil is going to actually help, it's because, so who here has like ever played with a prism before? You all know. So when light passes in from one medium, like the air, into another, it gets bent. and different colors of light get bent different degrees. This is what causes the rainbow effect. Well, glass is one medium that it can go through, and then air is another. And then it's got glass again with the lens. All of this bending back and forth. So it's going through the glass here, and it gets bent a bit. And then it goes back into the air, and it gets bent more. And then it goes into the, the lens here, and it's going to get bent again. And some of the light is going to escape. It's not going to hit the lens because it's been bent so much. The difference between glass and air is much bigger than the difference between glass and oil. So here, it bends a little bit when it gets to the glass. It doesn't bend as much when it gets to the oil, and it still hits the lens. So the oil immersion is going to help more of the light actually reach the lens by reducing diffraction. So that was bright field microscopy, where you're looking at the shadow of something on a bright background. Dark field microscopy is, well, you're looking at bright objects on a dark background. How does that work, you ask? Or maybe you don't ask. Either way, I'm going to tell you. Basically, what happens is you have the light is not headed towards your eyes. You're looking down at it. The light's going that way. If there's no sample there, you look down and it looks black because all of the light's headed that way. None of it hits your eyes. But when the light's moving along, if it runs into something, it scatters. It bounces in a random direction. So anything that's there is going to look bright because you see the dark background and the light comes in, and let's say you've got one little thing right here. The light comes in, it goes boink, and some of it hits your eyes. So you're only going to be able to see the things that are there because that's the only bit that's going to be directing light towards your eyes. Dark field is going to have very good contrast. It's going to have somewhat less good resolution, but you'll be able to see things that you wouldn't be able to see with bright field. Phase microscopes are a little bit of a combination between the two, but they're mostly a type of bright field. They take advantage of the fact that light, which moves through a cell, like a bacteria, changes a little bit. And it changes because of refraction, what we talked about before with the bending of the light. Cells bend light differently than water does. So you've got one bit of light that goes through a cell, one bit of light that goes right next to the cell, and they come out slightly different. Phase microscopes are going to magnify that difference. And doing so, what they're going to do is highlight the boundaries. They're going to highlight the edges of objects and make those edges stand out. So here we see two light samples. They start off the same way. One passes through the bacteria, one doesn't. And you can see that they get a little out of sync. Like this is the, so this is the peak of this one, and it lines up with not quite the peak of that one. When it moves through what's called the phase plate, that difference is going to be magnified. So that now, actually, the differences are opposite. That when one goes up, the other goes down. And that means that these two light waves are going to be very easy to distinguish from one another, and that's going to increase the contrast of what you're looking at without staining it. Just to give you some pictures here, this on the upper left is bright field microscopy of some tissue cells with some bacteria inside of them. This is dark field microscopy, and you can see that these little dense bacteria, you can now see them very, very well. They just light up. But you lose a little bit of contrast. Like some of these things over here that you can definitely tell are two different objects. Over here, you might have two white dots with a little white bit underneath them as well, and it's a little bit harder to tell exactly. But you can definitely see them much easier. This down here This is a phase contrast image. So this is still bright field, but it's using that phase contrast trick. And you can see how it kind of makes the edges glow a little bit. So anywhere there's a phase change, anywhere there's a boundary, is going to be slightly luminescent. So you can tell where organisms stop and start with much easier. This is a standard phase contrast image. This is what's called a Normansky phase contrast image. It's using a very similar technology, but instead of making the outer edges glow, this is going to contrast the stuff on the inside by giving it a three-dimensional appearance. This is not a true 3D image. It's a trick of the light. And what looks like sort of is three-dimensional is it telling you this is where there's a boundary between something and something else. But it sort of looks like these are bacteria kind of floating on top. Yes?
Has there been like a preferred microscopy? Like out of these groups, is there any true one that has been preferred over the others?
Depends on what you want to do with it. But I would say for microbiologists specifically, if you're looking at bacteria and not like human cells, not really eukaryotic cells, big complex things, if you're looking at really tiny things that don't have super complex insides, most of them, they start off looking with phase contrast. And then they'll move to another technique if they need to. But I would say, for most bacterial applications, phase contrast is the default. This assumes that you're looking at unstained cells. If you're looking at stained cells, you want to be using a bright field. So there's also fluorescence microscopy. Fluorescence microscopy is very powerful. What you're going to be looking at is they're going to direct one wavelength of light across the sample. And that wavelength will be filtered out so that it can't get to your eyes. So you're going to shoot it with blue light, but you can't see blue. You can only see red. And there will be specific fluorophores, dyes, molecules, potentially labeling particular parts of the cell that are inside and that will glow when they're hit with the right wavelength of light, and that they will glow a different color. So you're hitting it with blue light, and then you have things inside of it that are going to emit red, and you'll be able to see the red light because it passes the filter. So sometimes you're going to direct UV light at the specimen, sometimes you're going to direct blue light. Either way, it's going to be at an oblique angle showing across it. And usually, you're going to use antibodies with fluorophores, with fluorescent agents attached to them to see the specifically labeled parts. But more and more these days, what we're actually doing is we're creating cells that have a fluorescent protein called GFP, or green fluorescent protein, genetically linked to the bit that we want to look at. So you can see here, these are antibodies with the dye on them, and these antibodies are to the cell surface of these bacterial cells, and they're going to coat the surface of the bacterial cell. And you can look at this bacteria, and you can say, whoa, that's definitely a nice contrast increase, right? No problem seeing the bacterial cell. And the really nice thing about this, as it's different from dark field microscopy, is with dark field microscopy, everything that's there gets lit up. With this, you could, for instance, have one antibody that labels E. coli. This E. coli could be in there with a bunch of staph and strep and whatever. And as long as those antibodies only stick to the E. coli, the only thing that you're going to see is the E. coli. Or you could label E. coli green and strep red and look at them next to each other. So, let's get started talking about staining. Hopefully most of you are sort of familiar with staining. What sorts of stains, those of you who are in the lab, what sorts of stains have you guys done at this point? Endospore. Graham.
Simple. Yeah,
Simple. That's yeah. And have you guys done a acid fast?
I'm doing that next week.
Next week. Okay. So hopefully some of this will be review. But so staining, as we said last time, is a method of increasing contrast in cells, and it is usually done in concert with microscopy. There's lots of different types of stains depending upon the types of organisms that you're looking at. A lot of them are specific for what we call histology, which is the study of usually human or mammalian tissues. So if you're a cell biologist or a human biologist, you're going to be looking at a lot of human cells that are stained with cells that will highlight particular organelles and such like. While that is of interest to us in microbiology, most of what you're going to be looking at are bacteria. So most of the stains that we're going to talk about today are stains that you would use in a situation with bacteria. So the first thing that you do, the first step to most staining procedures, is you have to get a thin film of the bacteria that can is usually made if you're just like taking bacteria from a lab setting. You just dilute it a little bit in water and let it dry. This is a smear. Most of you have probably done smears at this point. If you're looking at a tissue sample, and even as microbiologists, we may be looking at a tissue sample because there are some microbes that can't be cultured in the lab, and you have to actually look at them inside, from, if you're like a forensic microbiologist or something, and you're looking at bacteria that are living inside of someone, you would usually embed a piece of the tissue in wax, in paraffin, and make very thin slices of the wax, and then that's what you would look at. So all microbiological stains contain a chromophore, which is basically just a colored thing. Although I guess there are fluorescent stains as well. Usually we refer to fluorescent stains as dyes, but they're effectively very similar things. Stains come in two varieties, acidic and basic. The vast majority of stains that are used in biology are basic stains or basic dyes. And in this case, basic doesn't mean simple. A simple stain is something that we're going to talk about in a second. But a basic stain or a basic dye means that it is not acidic, that it's alkali. It has a pH greater than 7. And generally speaking, things which are basic are positively charged. Things that are acidic are negatively charged. And what's your membrane made out of? Phospholipids. Who remembers the charge on a phospholipid? Or on a phosphate? Negative. The reason why the vast majority of stains that we use in microbiology, or any sort of biology to be entirely honest, are basic is because usually what we want to be looking at is something with a membrane. And the membrane is going to stain very well with basic dyes. It's positive, and it's going to stick to negative things. Negative things like DNA is negative. The membranes are negative. Ribosomes are negative. Proteins could go either way, but you actually don't want proteins to stain most of the time because there's going to be proteins everywhere and just the whole stain will stain, or the whole cell will stain. Acidic dyes are used in a few specific circumstances for staining either basic structures or to get a negative stain, which we'll talk about in just a second. So there's basically two types, two general classifications of stains. Stains can be simple or differential. A simple stain is when you just want to look at something. You want to stain everything the same color. I want to look at cells. I'm going to stain all the cells purple. A differential stain always has two or more stains and is specifically for the purpose of being able to distinguish between two different things. The classic differential stain is the Gram stain. You guys should all be familiar with the Gram stain, whether or not you're taking lab. I'm not going to get too detailed into it. If you're not taking lab and you don't know what a Gram stain is, look it up. You'll need to know it. It'll be on the test. But the Gram stain is basically going to tell the difference between Gram-positive, thick cell wall bacteria, and Gram-negative, thin cell wall bacteria. And it's a differential stain. You have two colors on the same slide at the same time, and they're going to differentiate between two different things. So it's a differential stain. It helps you differentiate. Most stains that we use in microbiology that are like tests are going to be differential. The Gram stain is differential. Endospore is differential. Acid-fast is differential. Capsule stain is not. We'll get to that in a second. But the vast majority of stains are, because usually what you want to know is it this or is it that? You have a question?
These specific stains, are we going to need to remember these?
Not those ones. This is the one. This is a Gram stain. So these are to remind me if I wanted to talk about it. If you're staining, so histology stains, this is if you're looking at human cells in thin section, usually that have been embedded in wax, which I said you might do sometimes. GMS and HE are the two most common stains, differential stains to look at human cells with. With bacteria, probably your first go-to test is the Gram stain. So this is a nifty little Gram stain here. And you can tell the difference very easily. You've got nice, dark, purple cells on the left, and on the right you have sort of very light pink cells that are a little bit washed out in this particular picture. But you have nice light pink cells that don't take the purple dye. Or actually, they do take the purple dye, but it washes out. If there's one thing that you should all come out of lab knowing how to do, it's a Gram stain. Make sure you know it. So this is what's called a negative stain. Usually, a positive stain means that you are staining the feature that you want to look at. So Gram stains are a positive stain. You're staining the cells, you want to look at the cells. Capsule stains are typically negative stains. That means that you're staining everything except the feature that you want to look at. And you can see this is the cell stains. This is the background stains. That's the capsule. What's a capsule? Who remembers what a capsule is?
It's like this layer around the cell wall.
It's not the cell wall.
The layer
It's
around
outside
the
the cell wall.
Yeah.
Yes. It's a layer of basically snot, of mucus, of that surrounds the cell and protects it and serves it in attachment. And it's a virulence factor, right? Whether a cell has a capsule or not is often the difference between a pathogenic bug and a non-pathogenic one. So that's something that you want to know. But capsules are not positive and they're not negative. Charged. I mean, they're usually neutrally charged. So acidic dyes don't stick to them. Basic dyes don't stick to them. In fact, dyes in general don't stick to them, which can make them difficult to view directly. But here, you can use the fact that dyes don't stick to the capsule to make it so that you can see the dye sticks to everything else, except the capsule. So you can see the capsule by where the absence of the dye is. Is this a simple or differential stain? You say simple. Why do you say simple?
Because there's only one color. It's purple.
There's only one color. This is a simple stain.
How does the dye get into the capsule, into the inside of it?
The dye actually can move through the capsule just fine. It just doesn't stick to it. See, the cell is charged, so it sticks to the cell. The actually glass, believe it or not, is somewhat charged. It's silicon dioxide, and the oxygen in glass is a very easy thing for a lot of dyes to stick to, and so it'll stick to the glass just fine. But it'll move through the capsule, but it doesn't stick to it very well. It just doesn't get stuck in it. It would rather be anywhere else. Does that make sense? So this is a flagellar stain. Another special stain that you would see in some circumstances. Is this simple or differential? Simple. One color. With this, you're using specific special dyes that stick to flagellar proteins. And you can see all the flagella coming off of that cell. It's kind of nifty. When you look at cells most of the time, do they look like that? No. Usually you can't see the flagella. The flagella is not innately charged. Basic dyes don't stick to it. Acidic dyes don't stick to it very well either. You have to use a special dye that specifically will bond to the flagella in order to see it. So these are the steps of the gram stain. I wanted to say a little bit about what's going on in the gram stain really quickly. So the first step is that you add crystal violet. Crystal violet is not actually a basic dye. It's sort of a special dye. It doesn't stick to membranes. Most stick to membranes. So here we have a gram positive. Here we have a gram negative with a little membrane on the outside. Okay? Thick cell wall here. What happens is that the crystal violet gets inside. And this is, if you recall, sort of like a mesh. And the crystal violet gets inside and gets stuck in the mesh. And it does that in both cells. And you'll notice, if you do a gram stain, that actually they both start out looking purple. Second step is you add iodine, which is sometimes called Gram's reagent. Does anyone here know what that step is or what that reagent is for, what its job is? There's a special word for it. It's a mordant. A mordant, it's a word that you mostly see in like painting. But basically, if you think mordant, think mortar. What do you use mortar for?
Putting up a wall.
For putting up a wall. But basically, it's a cement. It's going to stick two things together. A mordant is going to be something that allows something to bind to something else. And what the gram iodine actually does is it goes in there and it crystallizes around the crystal violet, making it bigger. Next step, you decolorize. You're basically going to wash it in something that the dye wants to move into. In this case, it's acid alcohol. Now, the dye moved in pretty quickly because it was small. Now it's big, and it's going to move out slowly. So you decolorize, and the length of time for the decolorizing step is key. And you're going to get rid of the dye in the first couple layers here, and in the first couple layers here. The problem is that you've gotten rid of only the first couple of layers here. In this, it only had a couple of layers. So here, the wall is small enough that all of the dye can move out. Here, the wall is so thick, like these ones, maybe they moved a little bit up to here, but they didn't have enough time to actually get out of the entire cell wall. So this has still got the blue coloring, this doesn't. Then you hit it with safranin. Who can tell me what safranin does? Counterstain, what does that mean? Say it louder. You don't know? What's it going to stain? So, does it only stain gram-negative cells? It stains membranes. It's a normal, basic dye. Okay? And it's going to stain membranes. So it's going to stain this membrane pink. It's also going to stain this membrane pink. So why doesn't the gram-positive cell look pink? There's a thicker cell wall. It's still got the purple. This purple is a lot darker a color than this pink. So if you've ever looked at a gram stain and you said, God, why is that pink color so light? It's hard to see. That's sort of on purpose. I mean, not exactly on purpose, but it's helpful. Because that means that this purple is going to completely overwhelm the pink tinting of the inside. Whereas here, you see the pink because the pink is all that's there. Here's a little bit of a more differential gram stain there. You can see some pink bacilli and some purple cocces. So just, oh yeah, and here's an endospore stain. Sorry, my label got kind of covered there. Staining for electron microscopy. I think you guys talked about electron microscopy last time. I just wanted to point out, so the reason why you use colors to stain for light microscopy is because colors interact with light. What are you looking at in electron microscopy? Electrons. So would a colored dye do anything? What do you want your dye to interact with? What interacts with electrons? Metals. Heavy metals. Actually, it's nuclei. Any big nuclei. So you want to choose things from the bottom of the periodic table, which are almost always metals. There just aren't that many gases up there. So metals, heavy metals specifically, are what you're going to use. Like aluminum isn't going to work. Iron, just barely. But you get up to like uranium, osmium, lead, gold, silver, things up in that range. Mercury, those work well. They're going to block electrons. And what you're looking at is a shadow of the electrons. So when you stain for electron microscopy, you stain with heavy metals that stick to particular structures.